evening. My name is Katherine Xue, and I'm also a student at the University of Washington with the ENGAGE program. My research is I use computers and programming to study the unique fingerprints of molecules that we're going to try and find in space. And as a graduate student, I get really specialized training in how to do a very specific type of research. But the ENGAGE program gave me another type of training that graduate students very rarely get, which is how to communicate my science in a way that's engaging, enlightening, and informative. You can learn more about the ENGAGE's innovative program at engage-science.com. And now to our second guest. Katherine Xue is a graduate student in genome sciences at the University of Washington and the Fred Hutchinson Cancer Research Center. Today she's going to tell us about her work studying how the flu virus can evolve within a single person. Please give a warm welcome to Katherine Xue.
All right. Thanks, Meredith, for that really kind introduction. So I'm really excited today to have the opportunity to share with you a little bit about my research on flu evolution. So just to get us started, I just want to take a quick poll. How many of you guys think you may have gotten the flu in the past couple months? Show of hands. Yeah. See, I'm sort of in that group too, unfortunately. So we're coming to the end of a pretty bad flu season, both within the United States in general and actually, especially here in Seattle, where there were a large number of very severe flu cases. All in all, around the world, it's estimated that something like 1 billion people are infected with the flu each year. That is an enormous number of people, and what that means is that flu is a virus that really touches all of us in ways both big and small. As someone who studies the flu, I think about this a lot. I sort of encounter the virus in a lot of different settings. I grow the virus in the lab, I study it using computers, but just like everyone else here, I just sometimes get the flu, I get sick. And that's kind of the backdrop for what I want to talk to you guys about today. I'm going to tell you 3 stories from my work studying the flu. And these are 3 stories that take place at different times and different places, but hopefully you'll see by the end how all of them are really interconnected. So I'll jump right in. So part 1. Once upon a time, there was a person and the flu. And the flu being a virus, it did what viruses do best. It infected this person and it started to grow and grow. And as you can imagine, this person wasn't really very happy about this. Deep within the body, those immune cells that Hannah just told you about, they sprang into action against this foreign invader. Some of the cells identified the flu viruses and they swallowed them right up. And other cells threw out these little tiny tranquilizer molecules, antibodies, that would stick to the outside of the virus and stop it from being able to copy itself. And for a while, the immune system was able to start to get this infection under control. But this wasn't really the end of the story. Even as the immune system was doing its work, the virus continued to copy itself, until one day, a mutation happened. Now, mutations actually happen all the time. When I say mutation, I just mean some change in the virus. And the fact is, viruses are actually very sloppy at copying themselves, so mutations happen all the time. Most mutations are pretty bad for the virus. They usually break something. But in this case, this mutation was a little bit different. This mutation made a small change to the outside of the virus so that those antibodies that had stopped the other viruses weren't really working very effectively against this new virus. And that meant that over time, this virus started to grow and grow, and to grow and grow until eventually this person who had started out being infected with one flu virus was gradually taken over by this new mutant strain. And over time, through a cough or a sneeze, this new mutant virus started to spread from person to person. And it spread, until eventually it spread all the way across the world. Part 2. Once upon a time, there was a person and the flu. And this time, the person in the story is me. When I came here a couple of years ago to start graduate school, I was really interested in studying evolution. Evolution is the process of change that shapes all living things. But the problem with studying evolution is that it usually takes thousands or millions of years. And that was a lot longer than I really wanted to be in grad school. See some grad students in the audience. That's part of what got me studying the flu virus. Flu is a virus that changes incredibly fast, practically right in front of our eyes. Just think about the fact that, as you all already brought up, you have to get a new flu shot every year, whereas for viruses like measles or the smallpox, a single course of the vaccine can protect you for your entire life. As I started thinking about the flu, I started getting really obsessed with the story that I told you about earlier. That story was kind of a thought experiment. I really wanted to know how much could the flu virus be evolving within just a single person while they're sick. Now that might seem like a weird question to ask. Flu infections are pretty short in the scheme of things. I know it doesn't really feel that way when you have one, but they just last about a week. So how much change could possibly be occurring in that short of a time? But I thought that something must be going on, just because we know that flu is a virus that changes incredibly rapidly around the world. And all of that evolution has to start somewhere. Ultimately, it has to start with single mutations that arise within individual people while they're sick. You can think of it a little like this. We know that there's this complex global picture of flu evolution where strains are sort of changing from year to year across the globe. And we have a very good bird's eye view just from years of monitoring these flu strains. But I wanted to know what would happen if you really zoomed in really, really far down to the level of single people and their flu infections. What would you find there? The question that I eventually asked was this. What can one person's flu infection tell you about how the virus is changing across the globe? Now, this isn't a really easy question to study. It's actually really hard to try to measure how the virus is changing within people, within these pretty short infections. So we kind of had to come up with a different strategy. And this is where I got a little bit lucky, actually. One day, my advisor, Jesse, went downstairs to get a cup of coffee. And while he was there, he ran into two doctors, Steve and Michael, who also work at the Fred Hutch. And you know, they got to talking. Steve and Michael are like, hey, Jesse, what are you guys up to in the lab these days? And Jesse said, you know, we're actually starting to get interested in some questions that are sort of up your alley. We're starting to wonder how much the flu virus might change within a single person while they're sick. Is that something that you guys might know anything about? And Steve and Michael said, you know, as a matter of fact, yes. So they work with cancer patients at the Hutch who are often very vulnerable to viruses like the common cold or the flu. And they said this, well, we occasionally treat these patients who have incredibly long flu infections that last multiple weeks or even months. We're constantly taking viruses from these patients to try to understand if the infection is getting better or worse and if we should be changing what we're doing to treat these patients. It would actually be great if you guys wanted to go in and try to understand in more detail how these viruses are changing over time. So that night, I got an email that basically tells me the story that I just told you. There were these really unusual patients at the Fred Hutch who had had these very long infections. And by studying these patients, we might gain some insight into how the virus evolves more generally. And so I decided to propose to study this question in these patients for my thesis. And for those of you who have never had to defend a thesis proposal before, it feels a little bit like this. It's you at the front of a room in front of a lot of terrifying professors who ask these hard questions about what you're proposing to do and all of the many ways that it could possibly go wrong. So I think overall this went okay, just because I'm still in grad school. But there was one question that really stuck with me. And actually it came from my advisor, who transforms into a dragon for these occasions. He said to me, you know, Katherine, you're proposing to study these patients with very long flu infections. And the fact that these infections are so long is what makes it possible in the first place to study them. But you also have this idea that you want to learn something about how flu is changing across the globe. But what if these patients are just different? What if something really weird is just going on within these very, very long flu infections? What if you can't actually learn anything very broad and general by studying them? And I really didn't have a very good answer to this question. It seemed like something that we would just have to learn from the data. And so this was something that I filed away in the back of my mind as I really started working on this project. So that brings me to about last summer. I spent a lot of last summer working with the viruses that had been collected from these patients. To try to understand how the viruses were changing over time, we wanted to sequence the genomes of these viruses. So genomes are the instructions that encode for building all living things. And so changes in the genome cause changes in these viruses. By sequencing the genomes, we hope to get a really detailed picture of how these viruses were changing. But as soon as I started trying to do this, I started running into a lot of problems. It was just really hard to sequence the genomes of these viruses. Now, sequencing at this point is something that should be pretty straightforward. It's a little bit like following a recipe. It's something that seems like it should be very easy when it's working. But when things aren't really working the way you expect, you can spend a lot of time sort of fiddling with different ingredients and changing the protocol and just tweaking this step and that to try to optimize your experiments. I spent weeks and even months working on this. And as my experiments dragged on, I really started to have some doubts. I started to worry that maybe we would never actually be able to sequence the genomes of these viruses. These viruses were pretty old. They had been collected from these patients something like 10 years ago, and then they had been sitting in freezers in labs ever since. Genomes tend to go bad over time, and so I worried that maybe we would never get any data out of these samples. I also worried that even if we did sequence the genomes, maybe it would turn out that these viruses weren't changing at all. Maybe it was just my imagination that told me that there was some sort of change that was happening in just a couple weeks, and maybe I would just be proved wrong. But the doubt that sort of stuck with me the most was this question that my advisor had raised about whether anything that we found within these patients, within these very, very long, unusual infections, would actually tell us anything more general about flu evolution. And so as the weeks dragged on and my experiments still weren't working very well, all of these doubts really started piling up. And over time, I really started wondering whether maybe none of this had been a very good idea in the first place. I'm glad you guys think that's funny. Eventually there really wasn't very much left to do except to take this recipe that I had spent so long troubleshooting and to try it on these viruses and just hope for the best. All right, so this is part 3. This is the story of what we found. So once upon a time, there was a person and the flu. And this person wasn't just any person. Within this person, the cells that usually guard the body had gotten extremely sick, and they were starting to grow out of control. This person had cancer, and eventually there wasn't really anything left to do except to try to get rid of all of those sick cells and replace them with healthy new ones from another person. But there was a problem here. After all of the old sick cells had been gotten rid of, before the new ones really started to take hold, this person was extremely vulnerable. They didn't really have the defenses that we take for granted against viruses like the common cold or the flu. And that meant that when they got infected with the flu, the viruses quickly spiraled out of control. And the infection, which in a healthy individual would have just lasted about a week, went on for weeks, and then weeks became months. And what we found when we finally managed to sequence the genomes of these viruses, we found that these viruses were actually changing over time. They started out being mostly identical, but then over time, we saw mutations arising and then spreading within this patient. Here's just one example of one of those mutations, and it's colored in orange here. you can see that the mutation doesn't really appear until, at least not to detectable frequencies, until more than 2 months after the infections start. But then after it reaches this detectable frequency, it very quickly takes over within this patient. The name of this mutation is R292K. And for flu researchers, it's very famous because it gives the virus resistance to Tamiflu. This was a drug that the patient was taking to try to control the infection. And so this was an example of evolution that we could see basically right in front of our eyes. The viruses at the beginning of this patient's infection were very noticeably different from the viruses at the end. And when we started studying even just a single patient, we started finding many, many cases of mutations like these. And each of the mutations really had their own history. So here's one mutation here that didn't appear until very late in the infection, but then never reached a very high frequency. And that's pretty different from this mutation here, which was practically there from the start. And you can see that it eventually took over within this person. All in all, within any single patient in our study, we found several dozen cases of mutations like these. And each of them, again, had their own story. In some cases, like with R292K, we have some idea of what the mutation might be doing. But in most cases, we actually still have no clue. So I could stand here all night and tell you about the different mutations that we found, but I do want to get back to this question that I posed at the beginning about how evolution within individual patients related to this broader global picture of evolution. And you'll remember that the fatal flaw of this study was going to be that maybe these very long, unusual flu infections couldn't really tell us anything about what was happening across the globe. But when we started comparing the 4 patients in our study, we started seeing something really interesting. In several cases, the exact same mutation was arising in multiple patients, like this N225D mutation here, which we saw in 3 out of our 4 patients. Now, this is something that's extremely unlikely to happen purely due to chance. For those mutations to just occur randomly in the same place is something like the chances of winning the lottery. And so that clued us into the fact that there might have been something really strange and interesting going on. We suspected that this mutation might have something to do with helping the virus escape the patient's immune defenses. And we wanted to try to see whether other researchers had also seen this mutation in other flu strains across the globe. And when we looked in these global flu databases, we found that yes, in a lot of cases, the exact same mutations that we saw within the patients in our study went on to spread across the globe. Here's N225D again, colored in orange. You can see that it's present in a low percentage of flu strains from across the world in around 2006 and 2007 when the patients were infected. But then as of the last several years, it's really spread and taken over around the globe. This was incredibly surprising to us. It was not something that anyone involved in this study really expected. It was as though we had taken this global picture of flu evolution and zoomed into the level of single individuals. And then what we actually found was that these individuals recapitulated some of these same global evolutionary patterns that we were used to seeing. It seemed like in some cases, individual patients could actually act as microcosms of these global evolutionary patterns. So at this point, the story has pretty much caught up to the present. This is about where I am now in my work, trying to understand how flu evolves within single people. And so what's next? We really want to know a little bit more about how flu can change within the shorter infections that healthy individuals experience as well. Somewhere down the line, I hope that some of this information might be useful for people who are trying to predict what flu strains are arising and which ones may be particularly harmful. I do have to admit that that is a goal that's pretty far down the line. Everything that I've told you about today is just one tiny piece of all of the flu research that's going on around the globe. You could even say that it's just one pixel. But at least for now, being part of that pixel is something that's made me very happy. So I want to wrap up with a short epilogue. And as you might have guessed, this is another story about a person and the flu. And this time, the person in the story is you. I wish I could say that this was an entirely made-up story, and no one in this room would ever have to encounter the flu again. But the fact is, all of us have been and will continue to be characters in this story. And so I hope you think about that next time you get the flu. I hope you think about the fact that you're just one part of this broad evolutionary story that stretches across years and decades and around the world. And I hope that you also stop to think about some of the tiny little evolutionary stories that may be starting to happen within you. I want to thank all of the people who are involved in this study, especially my advisor, Jesse. I'm really lucky to work in awesome departments at both UW and the Fred Hutch, and I'm very lucky to be funded by both the National Science Foundation and the Hertz Foundation. And special thanks especially to Engage in Town Hall for letting me share my research. And of course, thanks most of all to all of you for your time and attention. And I'll be happy to take questions. Thanks.
So I was just wondering, do you think that the mutations that were there were just a result of a lack of an immune system, or just kind of a natural evolution from the starting point that they kind of came into?
Yeah, that's a great question. How much of these, how much of the mutation that we observed was sort of a response to these patients' sort of unusual immune status? Initially, like I mentioned, we were really suspecting that there might be mutations that were very specific to the context of these patients. But the fact that we see a lot of these same mutations arising in healthy individuals across the globe suggests to us that they're probably playing a more general role. But yeah, that's something that is that's very hard to disentangle.
So the question of the hour is, how do the researchers or maybe centers of disease control each year determine what vaccine to use? And is it mostly a recreation of what had been used in prior years, or something new and different each year?
Yeah, that's a fantastic question, and it's a very complicated process. So what I understand of it is that there are these surveillance centers across the globe that are constantly collecting flu samples from patients, and so they're sequencing them constantly to understand how the virus is changing. Every so often we'll see that sort of new viruses carrying new mutations are arising, but the really hard part of trying to pick a strain to include in the vaccine is the fact that at any given time there may be sort of multiple different new strains of flu that are all there, maybe making up like 20 to 30% of the population. And so the really difficult part is taking those different flu strains and trying to predict which of those 2 or 3 is really going to become the dominant strain in the next year. And of course, just in order to be able to make all of these millions of vaccines, they have to make this decision pretty early. For our flu season in the Northern Hemisphere, they're making this decision in maybe about February, whereas we wouldn't get the vaccines until about September. So as for how they actually choose which strains to include, they're usually looking, keeping an eye out for strains that are antigenically different from previous strains, and so they can test these strains in the lab and see if they have different immune properties from previous strains. The idea is that strains that seem really different from everything we've encountered before are likely to have a bigger impact on humans. And so that's one criterion. And then people have developed all kinds of really complicated and interesting models to try to explore this question. But it is something that's really hard to do.
A related question, because I understand it does take a long time to actually grow the vaccine. Can you speak to what's being done to shorten that cycle so that we can respond more quickly when we discover a particularly virulent strain?
Sure, absolutely. So I think there are a lot of steps in this vaccine production process that people are trying to speed up. One is just sort of open sharing of the data. And so these strains are collected from all across the globe. And you can imagine that, you know, if there's a sort of several month lag in sort of posting the data so that other researchers have it available and can sort of make predictions, then that can sort of really put us behind in our predictions. So that's one aspect of it. I think people are constantly exploring new methods to be able to produce vaccines at much larger scale. Currently, they're primarily produced in eggs, which is why if you have an egg allergy, you can't get the flu shot. And so certainly there are sort of other technologies being explored to try to speed up that process as well. Yeah.